Hi everyone, teacher Janet here. So this is the final video for the year, and it's about SPM paper 2 for those who are taking the paper this year. So this may be helpful to you. 10 winning strategies to answer structure and essay questions. This is the last part, which is part 3, where I'll discuss certain types of questions and how to analyze and break them down, and also how to get more marks through the answering of the questions and through the strategies. All right. So I'll be giving you some new questions also to try. All right. And some are concerning chapter 7 of Form 4. Chapter 7 has not been asked before. Okay, cellular respiration, Form 4. So there are around 11 questions, and the time is quite long, 1 hour 27 minutes, around there. So if you don't have time to finish all the questions, it's okay lah. It's up to you. But try at least the chapter 7 questions. All right, which there are a few questions there, chapter 7. All right, and the other questions, actually they are quite good questions. So see how you can, whether you have the time to do it. If not, then don't stress about it. All right. Okay, so let's get started. Okay, so we're going to look at different types of SPM questions and then the strategies to answer them. So here's a wise proverb. Give a man a fish and you feed him for 1 day. Okay, but if you teach a man the fish, you feed him for a lifetime. Okay, he'll be able to eat for a lifetime because he'll keep fishing, right? You'll be able to have a supply of fishes. Now, so the lesson learned from it is don't just aim to acquire facts or learn facts, right? To try to get marks by learning facts and then just remembering them and then applying them. But also learn the answering techniques and strategies to help you fish for more marks in exams. Okay, so here are some learning techniques and strategies to help you get more marks. And this is very useful for SPM biology questions because I geared towards the answering of biology paper 2 questions, all right, which has a specific mode of asking questions. So these are the different types of questions. There are about 10 different types that I can identify. Okay, the first one is labeling, state the name, definition, function, example, characteristic, adaptation. All this has been discussed in part 1, and we can memorize these details from the textbook and take note of the keywords used to describe them or used to answer them. Okay. So secondly is to draw and label the diagram. Your diagram must be biologically correct with complete lines, no gaps, and neat and labeled. All right. So that I've put as part 2 for Form 4 diagrams. Right. Now next is increase or decrease, more or less, not compare with original mode. So when to use increase, decrease, reduce, more or less, or not. That means comparing a process with the original mode. Okay, now this one we are going to discuss as we go along for this video. Then event, process, and final effect result. So a lot of the questions will use this kind of structure. There will be an event, and then they'll ask you for the final effect on something, and then after that ask you to explain. When you explain, that means you're actually explaining the process that causes the effect. Okay, so we'll discuss this. Explain effect of A on B, also a very popular question. Predict what happens. I couldn't find another question on this really, so we'll skip this, but I think I put some videos on prediction in the previous trial papers. Okay, comparing diseases or just explaining about disease. So to prepare for this, you have to know the definition, cause, symptoms, effects, prevention, and treatment of all the diseases. All right, and many diseases have been asked in the exam already. All right, such as one of them is thrombosis, embolism, COPD, on the diseases of the respiratory tract, and so forth. Calculation question. Okay, memorize all the formulas for quadrant sampling technique, CMRR technique, capture, mark, release, and capture technique. And I've one calculation for inheritance topic right at the end of this video. Okay, so no time to do all this already to give you a thorough explanation of this. All right, I'll just give you an example of calculation using calculation of probability. Okay, Now, then we have the comparison question where you compare two processes or two structures, okay? And then we find out what are the similarities and differences between them. So this is very popular. Every year also got this kind of comparison question, okay? So for similarity, you write both have this, and then for differences, you write A has this whereas B does not, okay? So one example is, which I've shown inside here, is comparison between Alzheimer's disease patient and Parkinson's disease patient, and also comparison between aerobic respiration with lactic acid fermentation. Okay, so these are good questions to try. Now, so compare different aspects such as the name of the process, the organelle, cell, tissue, organ, or organism involved, the structures, compare the structures within the structure itself, and then effect, process, where does it occur, the location, aim, function of the process, and so forth. Okay, so this will also be discussed later. And 10 is a hot question, how to tackle new questions systematically to score, even if you haven't learned it before. Now, but I have not arranged the questions according to this order, okay, because I already started doing the videos and then the slides, and then it's very difficult to rearrange it. All right, so we'll just number it 1, 2, 3, 4, 5, 6, and so forth. All right, as we discuss the different types of questions. So for our start, we'll look at some of these important strategies. The first one is using increase, decrease, more, less, or not mode. That means we are comparing the condition with the situation with the original situation mode. Okay, so this is a very popular and important question. All right, and I've already given many examples of this in the also in the SPP paper and molecular paper. Okay, then the event to process to final effect and result. So also mentioned in other papers also. So I didn't repeat the papers here. Okay, so state the effect or result first, and then explain the steps in between that comes after the event. All right, and that leads to the final effect. So we'll discuss this in a little more detail and explain effect of A and on B. It still has the same structure of event, process, and final effect. Okay, so it's the same step as the above. State the end result or effect first, and then explain the steps in between leading to the end result. Okay. Now, the sequence of the different types of questions will not be following the slide at the beginning already, because I have arrange it in a different way, and it's very difficult to reorder it again. Okay, never mind. You just follow the title, okay, on this that is explained to you here, and then we'll look at the questions. Now, what is the strategy to answer this type of question? Where you have an event, process, and effect. All right. So, for example, now this was in the SPP paper 2, which I posted today. Explain the effect on the characteristics of an organism if there's a change in the base sequence in the DNA molecule. Okay, so as an example, when you read the question, identify the event. The event is the first thing that occurs, is the change in the base sequence, ATGC, adenine, thymine, guanine, cytosine, in the DNA molecule. Okay, then you find out what effect they want you to talk about. So effect on the characteristics of the organism, from the aspect of characteristics of organism. So state this effect first. Okay, now as you can see here, first identify the event. Next, process, step A, step B, leave that first. The effect is in the question. So secondly, to answer, state the effect on something first. Like for example, state the effect on the characteristics of the organism. Then only you come back to explain the process step by step. How did this event lead to the process and lead to the final effect? Okay, now I'll give you an example of this. So if the base sequence of the bases in the DNA molecule is changed, that means instead of ATG it becomes ACG. Okay, that means the characteristics of the organism will be changed. That is the effect, the final effect, because it asks you to give effect on the characteristics. So you mentioned characteristics of the organism will be changed. Okay, then after that you only give the reason. What happens? So when the event occurs, mutation occurs. See, so this is because mutation occurs. You go back to the beginning. When the event occurs, mutation, that means spontaneous random change in the genetic material. That means the basis has occurred. Okay. Next, what is the next process that happens? Cause and effect. Okay. So when the mutation occurs, the genetic code used to synthesize the protein is changed. The genetic code is the triplet, the base triplets that symbolize or that give the code for the amino acid to be joined to the protein. Okay, the genetic code is the base triplets that is the code for the type of amino acid to be joined to the protein chain or polypeptide chain. Okay, so when it's changed, that means it will cause changes in the amino acid sequence in a protein structure, right? That's formed. So abnormal and non-functional protein will be produced. So notice that these are all the steps of the process. One step leads to another. Okay, cause and effect. And finally, that's why the characteristics of the organism is changed. Okay, so if it's 3 marks, 1 mark will be for P1, and then another 2 marks will be for the rest. Okay, any one of the rest. So always identify your event and effect on what first, and then answer the effect first, and then go backwards and explain what the process is about. Okay, so the first question is about the compensation point from 5 chapter 2, right? So let's read. Diagram 3.2 shows the effect of light intensity on the intake and release of carbon dioxide by plant. Okay, so here's the 0 point. It means that below the 0 point, during the change in light intensity throughout the day, below 0 point means that the lower you go, there's more release of carbon dioxide into the atmosphere. Why? Because down here below 0, respiration, the rate of respiration is more than the rate of photosynthesis. Okay, so the equation for respiration is go wet, and the equation for photosynthesis is wet go, just the opposite. All right, so if respiration occurs faster in darkness, right, when there is no photosynthesis, then more carbon dioxide will be released. Okay, but once the light intensity increases, there will come a point where the rate of photosynthesis and respiration is the same. That means whatever carbon dioxide is released in respiration is used in photosynthesis, in the synthesis of organic food substances. Okay, that's at Q. All right, so there's no intake of carbon dioxide from the air, neither is there release of carbon dioxide from the air, from the plant. Okay, no release of carbon dioxide from the plant at Q. Okay, whatever is produced in respiration is used in photosynthesis. Okay, so the plant doesn't exchange any CO2 with the atmosphere. Now beyond that, that's what we want to know. Okay, now beyond that, the photosynthesis rate, the rate of photosynthesis will be higher than the rate of respiration already, because there's more and more sunlight already. Okay, then what happens is that the intake of carbon dioxide will increase. Okay, because carbon dioxide is used in photosynthesis. All right, so carbon dioxide is taken from the air and used to manufacture or produce glucose. All right, produce food substances. So based on diagram 3.2, explain the effect on the production of glucose and the release of oxygen by the plant when light intensity increases. Again, let us identify our event, which is light intensity increases, right? And then you ask to explain the effect on what? Two things: production of glucose and release of oxygen. So you must mention that this at the beginning of your answer. Okay, you must mention and say the production of glucose will increase or decrease. It should increase, right? And the release of oxygen through photosynthesis will also increase. Okay, remember wet go is for photosynthesis, GO, wet go. So the O there is for oxygen. Oxygen is released. So the higher the rate of photosynthesis, the greater the amount of sunlight, the more the amount of sunlight gets brighter, then the higher the rate of photosynthesis, and then the more the release, the release of oxygen will be even more, okay, through photosynthesis. All right, so let's write the answer down. So you have to identify the event, and after that you have to state the effect on these two factors, and then explain the process. Okay. Now, so let's see. Now this is the same question. So answer, the rate of production of glucose, you see, we are talking about the rate of production of glucose after the point, the compensation point Q. It will increase and exceed the usage of glucose in respiration. Production of glucose through photosynthesis will increase and exceed the rate of usage of glucose in respiration. Okay? And then for the release of oxygen, more oxygen will be released by the plant to the environment. Okay? Through the process of photosynthesis. Okay, more oxygen will be produced. Wet go, think of wet go. Now next, so you continue to explain, because the rate of photosynthesis becomes higher than the rate of respiration after point Q. Okay, so these things will occur. And more carbon dioxide is taken up by the plant from the atmosphere for photosynthesis again, right? So then the carbon dioxide is used to synthesize the glucose, right? So excess glucose is produced and stored as starch in the plant, all right? So it's 3 marks. So 1 mark for rate of production of glucose increases, 1 mark for more oxygen, and the last mark can be for P3, P4, or P5. Okay, so your strategy is to identify the event, which is light intensity increasing more than Q, then state the effects first on the glucose production and release of oxygen, and finally you go back and explain what steps occur after the event occurred when light intensity increases beyond Q. What happened to the process here? Step by step you explain. Okay. Now what we want to discuss is this one. Okay. Mr. R was involved in an accident, okay, and his doctor found out that the neuron X was injured. Neuron X here, okay, was injured. Explain how this injury affects the knee-jerk reflex of Mr. R. This is a hot question, okay. So identify the event. The event is the neuron X was injured, okay, and then what is the effect that you need to state? The knee-jerk reflex. What happens to it? Okay, and on top of that, you can first identify what neuron X is. Okay, so the answer, X is the sensory neuron. When X is injured, it cannot transmit nerve impulse to Y, the motor neuron. Okay, because it's already injured, so the impulse cannot flow, right? So after that, Y itself cannot transmit impulse to muscle here. Okay, so also called the quadriceps muscle or effector. So muscle effector cannot contract. This one. The lower leg cannot jerk forward. Okay, because of the damage to the sensory neuron, neuron X. So our strategy here, okay, look below here, the strategy is to state P2 to P4 as the process, right? And you activate the NOT mode. You cannot transmit impulse, cannot contract. Okay, I've put HOTS question as number 6. So we're not following the system for the types of questions as at the beginning of this video. All right, you won't follow that sequence already. I'll just number it as it comes. All right, because I already put this HOTS question at this position, so it's very hard for me to shift all the slides to the right position again. All right, so we'll just take this as number 6, and whatever comes next will be number 7 and so forth, whichever type of question that we discuss. All right, now for HOTS question, how to tackle a new question systematically to score, even if you haven't learned it before. Well, you have to look at the clues in the diagrams and in the information given, and then ask what chapter is it trying to test you, the question, what chapter is it from, and then try to recall the content of that chapter and also the keywords, and try to apply what you have studied in that chapter for that situation. Sometimes it's not that hard to answer the question, but we have to stay positive and try our best, okay? But based on the hints and the clues, with enough training, you should be able to at least score 70 to 80% of the marks for HOTS question, right? Question 3. Diagram 11.1 shows one method to assist the growth of a plant seedling. All right, that is by planting the leguminous plant with the root nodules. All right, so this is about the nitrogen cycle. All right, if the legume plant was attacked by disease and died, explain what happens to the growth of the seedling. Now, so you have the event, the legume plant attacked by disease and died. What happens to the growth? So growth of seedling is your effect. You're going to talk about that. All right, and after you talk about the effect, then only you state or explain the step-by-step process of what happened after the legume plant was attacked by disease and died. Okay, so let's look at the answer. Now, the first thing is to talk about the growth of the seedling as mentioned in the question. Always follow the question. So the growth of the seedling will be reduced or stunted. All right, and then you go back to the event. When the leguminous plant dies, what happened? First thing, Rhizobium species bacteria is actually living in the root nodule. But when the plant dies, it has no more shelter. Cannot live in the plant roots anymore already. Okay, the plant roots have withered and dried up. So there will be less Rhizobium species in the soil. Okay, and what is the function of Rhizobium? It fixes the nitrogen from the air. So the nitrogen fixing process is reduced, meaning it helps to convert the nitrogen in the air into ammonium ions, but now the nitrogen in the air cannot be converted to ammonium ions because there are less or no rhizobium in the soil, right? They need the root nodules to grow inside. Okay, without the root nodules, they will die. So now we'll talk about the nitrogen fixing process being reduced. Now, actually, after that, what happens to the ammonium ions that are formed? They will be converted to nitrite and nitrite to nitrate. Okay, but then because of the reduction of nitrogen fixing process, then the nitrogen in the air cannot be converted to ammonium ions, so there'll be less ammonium ions. Don't say no, I'll just say less ammonium ions are converted to nitrite ions by what? By the nitrifying bacteria called Nitrosomonas. Either nitrifying bacteria, the general name, or the specific name is Nitrosomonas species. Okay, and then from nitrite, it has to be converted to nitrate, but there are now less nitrite ions, okay, converted to nitrate ions already. Okay, so it's a cause and effect kind of reaction process. Okay, one reaction will lead to the other, right? When there's less ammonium ions, then there'll be less nitrite ions. So less nitrite ions are converted to nitrate ions by again nitrifying bacteria, but different type of Nitrobacter species bacteria. Okay, then from nitrite ions, actually after converting to nitrate ions, the nitrate ions are absorbed by the plant seedlings. Okay, when it's produced in the soil. But if there are less nitrite ions, there will be less nitrates absorbed by the plant seedlings. Okay, so write the word less all over. This is a comparison that you have to make. Okay, because you're comparing with the normal nitrogen cycle. Okay, so soil becomes less fertile because of less nitrates, and then the plant seedlings will synthesize less proteins using this small amount of nitrates, okay, in the soil. So it will synthesize less proteins for growth. That's why the growth of the seedlings will be stunted. Okay, so you can see that what you have mentioned here is a reduced version of the nitrogen cycle. Everything is reduced in the nitrogen cycle. You already know the actual nitrogen cycle. You'll be able to write this very well. Okay, by using less like that, or reduced, or cannot. Okay, so our guide here is that steps or strategy here is state the final effect first based on the question, which is P1, growth of seedling is reduced or stunted. Then activate the comparison mode and the not mode. So you use reduce, less, not lesser, less, reduce, and so forth. And then activate the not mode, but cannot. Okay, and then explain step by step the process after the event has occurred, which is P2 till P9. Okay, the event that occurred, but step by step. Okay, all right, and then the process is a reduced version of the nitrogen cycle. Now here's another hot question. A group of scientists have done a study by injecting Wolbachia bacteria into Aedes aegypti mosquito eggs. This study is said to help and in environmental sustainability and is an innovation to help deal with the increase in dengue cases in Malaysia. Explain why. Okay, so you have to take up, it helps in environmental sustainability. How does it do it? It's an innovation to help in dealing with the increase in dengue cases. That means it must bring down or decrease the dengue cases. Okay, so Wolbachia bacteria is also discussed in your textbook in one of the side topics. All right, in chapter 8. Okay, you can look it up if you have time, but just a little bit. So you have to use your brains to connect it all together to get your 3 marks. Okay, now let's see how to do it. So first you must mention the effect of the Wolbachia bacteria based on textbook. All right, it is the bacteria is able to slow down or curb the growth of dengue virus in mosquitoes. Now actually the dengue virus, it doesn't really grow, but it can replicate. So maybe it's curbing the replication or reproduction of this dengue virus. Okay, so the virus cannot replicate in mosquitoes when the Wolbachia bacteria is inside the mosquito. Cannot replicate well. So that means it is in small concentrations and it will not be transmitted to humans through the mosquito bites. Okay, once the mosquito got the Wolbachia bacteria, right? So this prevents the spread of dengue virus among humans because the virus cannot replicate in the mosquito. It's not transmitted to the humans. So it will decrease the dengue cases. You see, we have to talk about dealing with the increase in dengue cases. Okay, so the Wolbachia bacteria will decrease dengue cases. This method does not use chemicals, insecticides, or pesticides, which kills other organisms. So it's considered to be environmentally friendly. That is to help explain this part. Okay, the bacteria helps in environmental sustainability, right? So you have explained why and how it does it, because it does not involve chemical insecticides at all, just the bacteria to slow down the growth of the dengue virus in the mosquito. Okay, and lastly, when male mosquitoes carrying Wolbachia mate with females, the eggs will not hatch. Okay, somehow the eggs will not hatch because of the virus, because of the bacteria inside the eggs. Okay, so this reduces the mosquito population. Now, this is a question on environmental sustainability. Okay, so it's a hot question. All right, which you have to create your own answer, synthesis. All right, so it's a high order question. Diagram 1 below shows an ecotourism center built by Mr. R on an abandoned mining area. Ecotourism, what is it? So let's look at the meaning of ecotourism here, below here. Responsible travel to natural areas that conserves the environment. Okay, keeps the environment safe and takes care of it, protects it, sustains the well-being of the local people, okay, that can find jobs like tourist guide and all that, and involves education or awareness of environmental issues. All right, so that means the tourists who come and visit all these natural areas, but they have to be careful that to preserve, to protect the environment and not damage anything. Okay, not damage nature or cause any harm to the environment. So based on your biological knowledge, discuss the advantages and disadvantages of building an ecotourism area. What is the advantage? Okay, so you can talk about the aspects of protecting the local flora and fauna. All right, because the local flowers, plants, and animals, they are all not chased away. They are left there to live there as usual. And the tourists are the ones that have to be careful not to stress them out. Okay, so it raises awareness of environmental issues to those who visit the place, and it helps to improve the infrastructure facilities in the ecotourism area by providing through the building of roads, bridges, and also facilities. Okay, as more people come in, there will be more facilities, all right, for the people to use. So it increases job opportunities and income for local residents who can be tourist guides and sell crafts. So this will create a stronger economy, better economy. Okay, generates income funds also for research, right, and for protection of habitats. Now, disadvantages. Poor management can cause habitat destruction, stress on wildlife, Okay, pollution due to excessive visitors, and if the visitors are not under surveillance or guided, some of them may be quite willful to do something to harm, may harm the wildlife or the flora and fauna there. Okay, so tourists may face security problems because of encounters with dangerous animals from the forest, crime like thefts, petty thefts, or pickpockets, and can encounter health problems like slippery slopes and bridges, lack of emergency facilities, unpredictable storms and floods. Okay. Here's another hot question. The Ecotourism Management Officer has advised Mr. R to do some improvements to his ecotourism area so that the ecosystem area is more secure with environmentally sustainable practices, right? So this is the part you have to underline. Suggest improvements that he can do to make it more secure. So improvements to ensure ecosystem is more secure with environmentally sustainable practices. The first one is to install the closed circuit television for better security, detect and prevent thefts, crimes, and even damage to the ecosystem by some tourists, right? So this can all be detected. So this will discourage people from causing any problems, okay, in this area. Now, for environmental sustainability, educate visitors who come to reduce their waste products, stop using the plastic bags, right? The single-use plastic bags. Practice upcycling for environmental sustainability. For example, transform old items. Even the management can do that, okay, and into new high-quality ones. All right, that's called upcycling. So they can educate the public and the tourists on uh, practices that will encourage environmental sustainability. Okay, so minimize the waste by banning single-use plastics like plastic bags, bottles, spoons, all right, in the area, in the restaurants in the area and so forth, or in the in the cooking places and all that. So grow edible plants and fruit trees for organic produce and to produce clean fresh air, okay, from the plants, all right. And also eco enzymes can be used in the washing up of the floors and all that, okay. That's also part of uh, practices for environmental sustainability, okay. uh, and then use biological control to get rid of pests, for example rear cats to catch mice, and don't use mice pellets that are poisonous and so forth. Now, another hot question here on conservation. Diagram 9 shows a campaign poster that was put up for a conservation program of the orangutan, bongo species. Okay, the orangutan, their survival is in our hands. All right, so there is a campaign poster for conservation program. Justify the implementation of the conservation program. Why do you need to implement this kind of program? Okay, so again, it's from different aspects. Firstly, from the aspect of the orangutan. The orangutan is a critically endangered animal in East Malaysia, like in Sabah and Sarawak. Conservation programs prevent the extinction and protect the ecosystem. So the benefit to the orangutan is also benefit to the ecosystem, especially in the tropical rainforest, okay, which is the habitat of the orangutan. Population of orangutan are decreasing due to deforestation, palm oil plantations being built, and then poaching. Poaching means they are being stolen, right, from the forest and hunted down for some of them. Now, fourth point, conservation will ensure the survival of this species. So this is the benefit there. Then benefit to the environment, to the rainforest. Orangutans actually help to disperse the seeds. After they eat the fruits, they may have wandered off to another part of the forest, and then they'll just throw the seeds far away from the parent plant. So they help to disperse seeds, and they help to grow new trees, okay, when the seeds sprout and grow. So this maintains biodiversity and forest regeneration so that the forest trees can grow, new forest trees can grow, okay. Now, then let's talk about the benefit to the local communities, to the local people there. Ecotourism uh, can be practiced, all right, where tourists are allowed to visit the orangutan conservation center and to visit and observe the orangutan, okay, and in its habitat and see how they are fed the fruits and all that. I've been to one conservation center too in Sabah before. So it generates income for local communities, all right, through the when they act as tourist guides or sell handicrafts and so forth. So this is the benefit to the people there. And the conservation program helps to maintain a balanced ecosystem and preserve the food web. This is the benefit to the environment, okay? Because the orangutan is part of the food web, all right? So number 9, the program increases awareness about the endangered status of the orangutan, all right? This is education and creating awareness, promoting awareness of this is directed to the people, okay, and to the school children too. Now here's another question on environmental sustainability, okay, which can be asked in question 11 also. So daily news, this is the daily news. Certain states in Malaysia have fully enforced a no plastic bag everyday campaign since the end of year 2025. This means businesses no longer provide free plastic bags. Shoppers must bring reusable bags or buy alternatives such as the biodegradable bags to reduce waste. Okay, because these plastic bags, they are usually non-biodegradable. They cannot break down very quickly into simpler substances. So they will pollute the environment for a long time once they are thrown away. So, all right, what's the answer? All right, your question first. The question is, based on the information, suggest the effects or benefits or importance of implementing this campaign. Okay, the benefits towards the environment. 8 marks. All right. So let's look at the answer. Benefits of the No Plastic Bag campaign day. All right, is to help reduce plastic waste so that less plastic bags are thrown into the environment or into the trash bin and to be disposed in the landfills. All right. So that means there'll be less plastic waste and less landfills. All right. It also pollutes the river also. So to decrease pollution, like water pollution and soil contamination from the plastic bags, okay, especially water pollution when the black bags are thrown into the river. 1 mark. So another benefit is that if we reduce the number of plastic bag used, it protects marine life and prevents animals such as the turtle from ingesting the plastic. Some turtles think it is a jellyfish, right, in the sea. So they will ingest or eat the plastic. And some will get entangled in the plastic nets. Okay, some fishes will get entangled. So this will cause the animals to die. Number 4, conserve natural resources. It reduces energy because fossil fuels is used for production of plastics, actually, right? So if you reduce the use of the plastic bag, it will reduce the energy and fossil fuels used for production of plastics. Then number 5, encourage recycling. So by forbidding the use of plastic bags, it will promote the use of reusable bags and eco-friendly alternatives like the biodegradable plastic bag. The plastic bag can be broken down quickly, okay, within 1 or 2 weeks into simpler, harmless substances. So those are biodegradable bags, but most are the plastic bags which are non-biodegradable. Okay, most of plastic bags that we use. Prevent clogging of drains and sewers by discarded plastic bags that can cause floods. So if these plastic bags land up in the drain and in the sewerage system, then it can block the flow of water and it can cause floods. Number 7, improve public awareness, okay, to educate society on environmental responsibility. Number 8, support sustainable lifestyle, encourages long-term eco-friendly habits among the public. Okay, cleaner environment, reduces litter in the public spaces, rivers and oceans, and it helps to reduce climate change. How? Because there will be less plastic burning. So this will reduce the greenhouse gas emissions. Okay, when the plastic is burned, it will release the gases that can become greenhouse gases to trap heat in the environment. All right. So if it's 8 marks, then any 8 of this. So the strategy to answer benefits of environmental programs. How to answer? Always think from the benefits of the environment. What's the benefit to the environment if we implement no plastic bag day? Less water pollution, less air pollution through the burning of the plastic bags. Then you reduce the waste in the landfill, okay, so less waste in the landfill. And benefit to the wildlife is to protect the plants and animals and also to protect biodiversity, okay, so that there are various plants and animals of different species on the earth. Now, humans, what's the benefit to humans? Create awareness, educate public to protect the environment. And there'll be less toxins if you use less plastic, okay? Less toxins will leach into the soil, or or less plastics will enter our containers, all right, which may contain microplastics, all right? So less toxins means better health. Energy resources to conserve the petroleum resources, less burning of fossil fuels, less greenhouse effect, and less global warming, okay? So the plastic is actually made from some compounds related to petroleum. So let's see how we can get more marks even though we are not sure how to answer a HOTS question, right? Now, cystic fibrosis is a hereditary disease. It is caused by mutation in the gene that produces cystic fibrosis transmembrane conductance regulator, CFTR protein. Okay, so when there's a mutation in the gene that produces this CFTR protein, that means the protein produced becomes abnormal. Okay, so the mutated CFTR gene fails to produce a CFTR protein that is functional. All right, and because the protein is not functional, the person gets cystic fibrosis. Okay, so now there are steps involved in treating the disease. Now let's see what happens here. So when you look at this set of questions with number 1, 2, 3, 4, 5, try and describe each step. That's all you can do. Describe each step. For example, first you have a virus that is non-virulent. So you can say, first of all, say that you prepare the non-virulent virus, right? If you know the technique, you can even state that at the beginning. So the technique is what? It's actually gene therapy. Okay, but even if you don't know, you can still answer. Okay, it's how many marks is this? I think this one is about 3 to 4 marks. Okay, so you just say prepare a non-virulent virus, and then look at the second diagram. The gene CFTR, the normal one, normal CFTR gene, has been placed inside it. This is the DNA, right? Has been placed inside the virus. So you say insert the normal CFTR gene into the virus. State that, okay? Because they ask you to. Okay, the question is describe the steps involved in the treatment procedure of cystic fibrosis. All these steps, based on this diagram, you're supposed to describe the steps involved in the treatment of the procedure of the treatment of cystic fibrosis. Okay, so first you mentioned gene therapy is used to treat the disease. Then a non-virulent virus is prepared. Okay, then insert the normal CFTR gene into it. Now you can mention that the virus is a vector, is good, but if you cannot, never mind. So after that, the normal CFTR gene is inserted in the virus already. Then the virus will settle on the cell and it will insert or inject the normal CFTR gene into the patient cell, as what you see here, right? So just write the steps down here first and then briefly, and then you can write your essay answer or structured answer. Okay, so once it's inserted the normal CFTR gene into the patient cell, this normal gene will replace this one, the red one, which is abnormal. It will start to produce the protein. The cell will start to produce the right protein based on the gene that has been injected into it. So the normal gene will replace the abnormal gene so that the cell is able to produce the CFTR protein is produced by the cell. See? So all your answer you actually get from the picture. You just try to explain the picture from step 1 to 2 to 3 to 4 and so forth. Okay? Now, just as a side note, in the textbook it mentions that gene therapy is used to treat genetic diseases. A normal gene like this one here is inserted into the patient to replace the abnormal gene, the red one here. Okay? Diseases that can be cured by gene therapy are cystic fibrosis, as mentioned here. Now, this is also true for muscular dystrophy and for cancer. Okay, the same process can be carried out. Now, the next type of question deals with comparison. Compare and contrast. Okay, so direct comparison question. Comparison between A and B. for example, between two processes, right? So we have to give the similarities and differences. Now this question can come out in the essay and also the structured question. All right. So similarities, both have something, dot, 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 dot. Differences, A has this, whereas B has and other things. So let's discuss the next question, question 9, based on Form 4, Chapter 7 on cellular respiration. Now, to remind you again, cellular respiration has not been asked in the exam yet, okay, in SPM for 4 years already. So it may be asked in this year, all right? It's the only chapter that has not been tested yet, okay? So let's read the question. The diagrams show two types of activities, A and B, which involve different methods to obtain energy through the breakdown of glucose in cells. Compare and contrast the two different processes of breakdown of glucose in the cells. Compare and contrast the two different processes of breakdown of glucose. So here we see a girl sitting down reading a book. She's not using a lot of energy, and she's carrying out what type of respiration? Normal aerobic respiration, okay, which goes by the equation GOWEC, the short form GO arrow WEC. Glucose plus oxygen produces water, energy, and carbon dioxide. Okay. Now this man is running fast. It looks like he's running fast. So he's carrying out aerobic respiration, but at the same time he's carrying out a different type of breakdown of glucose. What is that? Yes, it's called lactic acid fermentation in the muscle cell. Okay. So you're asked to compare and contrast the two different processes of breakdown of glucose in the cells. So similarities. Both these activities involve the breakdown of glucose into pyruvate in cells. Okay, and that is the first process called glycolysis that happens in the cells. Okay, to produce some energy, a little bit of energy. So both processes begin with glycolysis in the cytoplasm of cells, and both processes produce chemical energy in the form of ATP. All right, but the number of ATP for activity B produced through activity B is quite small compared to A, okay? Because A has more oxygen, right? Activity A. So this will cause breakdown of glucose to be faster, okay, and more complete. Never mind. Let's go on to the differences in the next slide. All right, so the differences. Activity A involves aerobic respiration, the girl reading the book. Activity B involves lactic acid fermentation, okay, for the athlete or runner that's running fast. Okay, so your process must be the right one. Okay, now next. Presence of oxygen for activity A, oxygen is present, but for activity B, oxygen is not present or is in limited amounts. All right. For A, it must be present in normal amounts. Okay. Complete breakdown of glucose is complete for activity A, but not incomplete for activity B. All right. That's why less energy is released in activity B. End products of process is water, carbon dioxide, and energy for activity A. Now we have to know the short form. GOWEC for aerobic respiration and gel for lactic acid fermentation. Okay. So I've discussed GOWEC now. For gel, is glucose breaks down to form E for what? Not ethanol. E for energy. Okay, a little bit of energy, and L for lactic acid. Okay. End products of the process. Compare them. So for activity A, aerobic respiration, the end products are water, carbon dioxide is C, okay, and E for energy. Okay, so this is aerobic respiration. And then for the end products of the process called lactic acid fermentation, it is gel, right? So that is E for energy and L for lactic acid. Okay, that's formed from the breakdown of glucose. So here water is not produced at all, and the lactic acid and energy is produced. Okay, but it is toxic, and later it has to be removed from the human muscles. right? Now, location of aerobic respiration is in the cytoplasm and mitochondria. Two places. Cytoplasm, where the glycolysis occurs. After that, it will move into the mitochondria, okay, where there's a reaction with oxygen. Now, so for activity B, it occurs only in the cytoplasm, okay. Mitochondria is not important. So energy produced per mole of glucose is large amount of energy for activity A and small amount for activity B, okay, because the glucose is not broken down completely for activity B, right? I can write the number of kilojoules per mole of glucose too, okay, but either you like write large and small amounts of energy, or you can also put in brackets. All right, so it's per mole, 2898 kilojoules per mole of glucose, not per molecule. Okay, one molecule cannot provide so much energy. So what's written in the textbook, I think is not that one. Maybe may have to be corrected. So actually, we are comparing 6 aspects: the product of the reaction or the process, the location where it occurs, the energy released, whether it's a lot or little. Equation, you can write down equation like these two here. Write it in full write it as a word equation. All right, no symbols but just words. Glucose, whether it's broken down completely or not, and O for oxygen, whether it's present or not present. Next, still on this topic of chapter 7, here's a new question. The microorganism above also carries out a process to produce energy through breakdown of glucose in the absence of oxygen. Take note, in the absence of oxygen. Okay. State the name of this process. First, identify what microorganism is this. Yes, it is yeast. So the name of the process will be alcohol fermentation. Word equation is actually GC. Okay, GC, not gel, GC. Glucose breaks down to produce carbon dioxide, ethanol, and energy. Right? That's why all your short equations, memory tips for equations are very useful. We have GOWAC, GC, and gel. All right. So glucose, this one is GC. Glucose breaks down in the absence of oxygen to produce ethanol plus carbon dioxide plus energy. Right? Okay. So word equation 1 mark. Now, word equation means you write it out in words. There are no chemical formulas. All right. For the different chemical substances. Number 3, compare and contrast the process in B, activity B, involving the runner, okay, with the process in B1 above. So now you're comparing different processes already. Process in activity B, we said is gel, lactic acid fermentation. Then you compare with B1 above, that's GC. So this is alcohol fermentation. You're comparing alcohol fermentation with lactic acid fermentation. Okay, alcohol fermentation, GC with lactic acid fermentation, gel. So since you're asked to compare and it's 3 marks, you must have at least one similarity and one difference. Okay, and then the other point can be either similarity or difference. Okay, so both processes involve incomplete breakdown of glucose. This is a similarity, right? Because both are fermentation processes. All right, and then in both processes, amount of energy produced is small. Okay, whether it's alcohol fermentation or lactic acid fermentation, the amount is only about 210 kilojoules per mole of glucose, whereas for aerobic respiration is much more. Okay, so the products for activity B, that means what products are produced, is lactic acid and energy. Lactic acid and energy. But for the process in B1, which is involving the yeast, so it's GC. So the products are carbon dioxide, ethanol, and energy. Okay, so two similarities, B1 and P2, and then 1 difference here on the products. Here is a hot question for chapter 7, cellular respiration. Why do athletes run or jog a few rounds after a race? After a race, right? Not before or not during the race. After the race, they will sometimes hold their flag and then run around the stadium, right now. So what's the reason? 3 marks. So it is a cooling down exercise after vigorous activity. Like when we have exercised very vigorously, you can also cool down by some light motions or movements. The jogging, very slow jogging, helps maintain faster blood circulation in the body. So it maintains the blood circulation because the heart is pumping faster to transport more lactic acid from the muscle to the liver to be broken down faster, to be removed. Okay, so lactic acid must go to the organ called the liver for it to be broken down into carbon dioxide and water. But it's found in the muscle, it accumulates in the muscle. So muscle must bring it to the liver. Okay, so if there's faster blood circulation, then more lactic acid will be transported to the liver. And it will not accumulate in the muscle. If it accumulates in the muscle, and there's no transportation or lack of transportation of the lactic acid to the liver, then the person will suffer from cramps, strains, and aches after a few days. Okay, so this prevents muscle cramps, strains, and aches later on if the lactic acid is removed. Okay, it allows the heart rate and breathing rate to return back to the normal situation gradually and smoothly. Very important. If not, there may be some harmful effects. Suddenly stop running. So questions on disease are quite popular. Sometimes in one paper two, there may be a few diseases that are asked, and also comparison between diseases. Okay, so we have to learn the diseases, facts about the diseases based on the definition, the cause, symptoms, effects, prevention, and treatment. These are the types of questions that may be asked in the exam, where you have to make a comparison. If you have to make comparison, then you just compare based on these, generally on these 4 aspects, okay, from these 4 aspects. And for comparison of genetic diseases caused by chromosomal mutation or gene mutation, we can compare based on, first give the name of the disease, the type of mutation, whether it's gene or chromosomal mutation, and then the process involved, and then the number of chromosomes in a diploid cell, okay, 44 plus XXY or 45 plus XY and so forth. Autosome and sex chromosome, which one is involved, and whether the patient is a male, female, or can be either gender. Okay, so process involved includes deletion, non-disjunction, what others? Insertion, base insertion, and so forth. All right. If it has the word base, like base deletion, that means it is a gene mutation. But if it's just chromosomal mutation, which is deletion, that is chromosomal mutation, not base. Okay, if there's no base in the front, that means it's the chromosomal mutation. So analysis of diseases that have been tested in the exam, SPM. So I won't read through all this, all right? So you can read through yourself if you want, and pause and read through, right? Now, for example, a comparison was made between osteoporosis and osteomalacia. So I think this one, many students were stumped by this question, all right? They don't know how to answer this. So you have to be ready for comparison of diseases, all right? Okay, let's look at this new question from Form 4, Chapter 12. All right, this is my own question. So it shows you 2 people who are facing health problems. All right, now the diagram shows 2 diseases related to the human nervous system. Compare and contrast between diseases R and S. All right, so in R, we have to interpret the diagram by looking at it. This man looks confused about the time or the location where he is, right? and his brain has shrunk, become smaller. Okay, diseased brain. So in this next picture for disease S, the person's hands have a tremor, shaking. Okay, and you can see the brain. It has neurotransmitter called dopamine, which has to uh, be transmitted to cross the gap here, the synapse. But in the normal brain, dopamine is a lot, but in the diseased brain, there's not much dopamine. Okay, so what's disease R and S? All right, so the diagram shows two diseases related to the human nervous system. Compare and contrast between diseases R and S. Okay, so from here we know that this disease is Parkinson, where there's tremor in the hand, okay, and the limbs, the legs also. Okay, tremor means shaking, it's shaking, always shaking. Whereas for disease R, is the motor movement of this person is still normal, okay, no tremors, but he forgets things, right? And he's confused about where he is. Okay, the thinking skills are involved. So this disease is Alzheimer's. Okay, I will teach you how to remember it in a short while. So similarities, both involve gradual changes or damage and death of brain cells. Both diseases involve gradual damage and death of brain cells. The brain cells are affected. And both diseases occur in older people around 50 years old. Okay, so in the elderly people. Now, for the differences, let's look at the next slide. Right, now let's look at differences between Alzheimer's disease and Parkinson's disease. So first state the name of the disease. It is not mentioned in the question. So disease R is Alzheimer's disease and disease S is Parkinson's disease. So what are the symptoms? This disease for Alzheimer's causes the loss of ability to reason, loss of memory. The patient becomes very forgetful. So look at the name, Alzheimer, think of memory loss. Okay. For Parkinson's disease, there's no memory loss. The patient will suffer from tremors in the lips, jaw, foot, or face. So tremors or shaking. So Parking, shaking. Okay, Parking, shaking, and Alzheimer, for memory. So it's different. The symptoms are different. Now, for Alzheimer's disease, the patient loses the ability to read and reason, but maintains body balance. Okay, that means he can still walk normally. But for Parkinson's disease, the patient has difficulty maintaining his body balance. Okay, his motor skills are also very bad. Okay, so he cannot maintain body balance well. Okay, like almost going to fall, but he has no memory loss. Okay, so Alzheimer's has to do with the brain, the mind. Okay, the loss of memory. Parkinson is more to do with the motor functions of the body. Okay, then lastly, this disease for disease R or Alzheimer's disease is caused by a lack of the neurotransmitter acetylcholine, okay, which has been cannot be produced because the cells that produce this neurotransmitter is damaged, the cells in the brain, okay, due to the abnormal protein deposits. So remember, it's connected to the lack of acetylcholine. But for Parkinson's disease, as you can see from the diagram earlier, is caused by a lack of the neurotransmitter dopamine instead. Okay, dopamine. Now, next question is from Form 5, Chapter 12, related to this chapter, which is on variation, all right, and genetic diseases. So it's a hot question. Diagrams 9.1a and 9.1b show the karyotypes of two individuals with genetic diseases. Compare and contrast between diseases A and B based on their karyotypes, which is a picture of all the chromosomes in one diploid cell. Okay, so they even circle for you the part that has a problem here. Okay, now look at diagram 9.1a. You can see that it has one chromosome number 23. What is chromosome number 23? Actually, it's the sex chromosome. In this case, it's the X chromosome. The larger one is the X chromosome. there's only one X chromosome. So remember, chromosome number 23 is actually the X chromosome. Okay, and also if, of course, if there's a shorter one, that's the Y chromosome. All right, in this case. So you can see that for the first karyotype A, this person has only one X chromosome. The X chromosome is a loner. So loner, remember, Turner. In internal, X is a loner. Okay, so this is Turner syndrome. Internal, X is a loner. Okay, it runs. Then for 9.1b, you can see 3 chromosomes number 21. So this is Down syndrome child, trisomy 21. Okay, now you're asked to compare and contrast between diseases A and B, so you must at least state one similarity and at least one difference. Okay, total marks is 6, so you must have a mixture of similarities and differences. 6 of them, 6 points. Okay, you get 6 marks. Now similarities, both involve chromosomal mutation, where there is a change in the chromosomal number. In both cases, there's spontaneous or random change in the genetic material or DNA in the cell. Okay, so this is the meaning of mutation. Now, both diseases can be caused by mutagens, which are substances that can cause mutation, such as food preservatives. In both cases, the mutation occurs during gamete formation. All right, that is the non-disjunction. In both cases, the mutation is non-disjunction, or rather, the process of non-disjunction leads to the mutation in the gametes first. Then, if the gamete is fertilized, then only it will produce the Turner syndrome child and Down syndrome child. Okay, let's look at the differences. Name of disease. Mention the name and you get 1 mark because it's not mentioned, right, in the question. So the disease is Turner syndrome for diagram 9.1a with 1 lone X chromosome in each diploid cell. And for B, 9.1b is Down syndrome. Take note that I've referred to the diagram as what they have written in the question, right? Diagram 9.1a and b. So number of chromosomes in the diploid cell is actually 44 plus X O, O or 0. So that means there's only 1 X chromosome here and 44 autosomes. Total number of chromosomes is 45 chromosomes. Whereas for diagram 9.1b Down syndrome, there are 3 chromosomes number 21. Okay, so the total number of chromosomes is the diploid number, which is 46, plus 1 extra chromosome number 21. So 47 chromosomes. Or you can write as 45 autosomes, okay, due to the extra chromosome number 21. There are 45 chromosomes instead of 44 autosomes. These are all autosomes. Plus, now if you look at this diagram, you will see that the sex chromosome is XY, one big, one small. So XY. So because it's XY, that means this is a male, right? So follow from the diagram in the question, all right? It shows the sex chromosome is X and Y, okay? Not XX. Now, cause. It is due to the lack of 1 sex chromosome for Turner syndrome. That is chromosome X, okay? But it's not mentioned here. But it's due to the lack of 1 sex chromosome X. For 9.1b, it is due to the presence of an extra chromosome number 21. Okay, as shown here. Gender of patient. For A, the Turner syndrome patient is always a female because she has no other chromosome, Y chromosome. Okay, so 1 X chromosome. This is for female characteristics, X chromosome. Okay, now for B, the patient can be a male or female depending on the sex chromosome, which is normal. It's just that there's an extra chromosome, 21. Okay, so in this case, he's a male. You can write them in brackets. Characteristics. The Turner syndrome patient has undeveloped breast and ovaries due to a lack of 1 more chromosome X. Okay, and has a web-like neck. For B, he has slanted eyes, flat nose, and broad forehead. All right. these are the symptoms or the characteristics that are different. Here's another question on diseases. This time is diabetes mellitus, which is a very common disease in Malaysia. All right. So it's from Form 4, Chapter 12 on homeostasis. Sorry, on coordination. All right. The endocrine system and the hormone insulin. So here we see lifestyle and dietary factor causes diabetes. You have to know how to read the diagram sometimes. All right. They'll give some diagrams that are like the flow charts. All right. So this picture shows a person testing his glucose levels. Okay. Now diagram 10 shows some of the factors that cause diabetes mellitus. Based on this diagram, suggest and explain preventive measures to reduce the risk of diabetes mellitus. 6 marks. Okay. So read the question carefully. Factors that cause diabetes mellitus, lifestyle and dietary factors. Okay, 2 of it. Suggest and explain preventive measures to reduce the risk of diabetes mellitus. So you have to suggest first and then explain. That means suggestion is 1 mark and explanation why is another mark. So remember, when you suggest and explain, all right, you will give steps plus the reason why. All right, this type of question has been asked before in the exam. All right, suggest and explain. So 1 mark for suggestion, 1 mark for explanation why you give this suggestion. Okay, and then you have to base it on diet. Dietary factor is diet and lifestyle. So cannot just write all on dietary factor. Okay, must have some points about dietary factor and some points on lifestyle. At least 1 for each one. Okay, from the aspect of diet. So you see, when you write the answer, you must clarify, is it dietary factor or lifestyle? Okay, now from the aspect of diet first, eat a balanced diet rich in fiber. This is the main thing, such as whole grains, vegetables, beans, and fruit. Why fiber? So this is your suggestion, and you must give the reason why. Explain. So this is the explanation. This is because high fiber foods help to control the blood sugar levels by slowing down the carbohydrate digestion. Stroke sugar absorption is also slowed down. Okay, so the soluble fiber will act like a gel and mix with the food, and will slow down the movement of food, so it doesn't get absorbed so fast in the small intestine. Okay, so it also slows down. Fiber also slows down the carbohydrate digestion, like oats. Oats is digested very slowly because it has a lot of fiber in it. Okay, now so you have suggestion. You can write it as F1, the fact, and then E1, the explanation. Must go together. Okay. Then you have F2, avoid. Fact means F means fact. Avoid sweet, sugary food. Okay. And then the explanation. These foods increase the blood sugar level faster. If you eat the sweet, sugary foods, it'll be the sugar will be easily absorbed into the blood. All right. So from the aspect of lifestyle, okay, only 1 suggestion. Carry out daily regular exercise, such as walking, to oxidize the glucose that you have that is high. The level of glucose that is high, you will oxidize the excess glucose in the muscle cells to release energy for muscle contraction. At the same time, the glucose is used up, so the level of glucose will decrease. So this reduces the blood glucose level. Okay, so 1 mark for each point here. So it's 6 marks, exactly 6 marks. Okay. So I've also decided to include 1 question on drawing of schematic diagrams for inheritance of traits, which is related to Form 5, Chapter 11 on inheritance. All right, how to draw the schematic diagram, especially for essay questions. So schematic diagrams involving inheritance of traits can be asked in the exam. All right, so there are many types of schematic diagrams. I just choose 1 as an example. So let's look at the last question. A man suffers from color blindness, whereas his wife has normal eyesight but is a carrier. X big B is a dominant allele for normal eyesight, while X small b is the recessive allele for color blindness. Okay, so we use these symbols afterwards. Draw a schematic diagram below to show the possible genotypes and phenotypes of their children. So inheritance topic so far they have not asked them to draw schematic diagrams yet in full. So get ready for it, all right? And we have to know all the different types of inheritance. Monohybrid, dihybrid inheritance, sex. This is about sex-linked inheritance, okay? Where the allele for hemophilia or color blindness is found on the sex chromosome, chromosome X, okay, or X chromosome. So we are supposed to draw a schematic diagram below to show the possible genotypes and phenotypes. Let's have a look. You can try it first also. Now, so the father, follow the description here. Father is colorblind. Okay, so father has the sex chromosomes X and Y, and then on the X chromosome there is the allele for color blindness, which is small b. Y doesn't carry any alleles for color blindness. Okay, so empty here. So for sex-linked disease, you must write the allele at the top here as a superscript. Okay, not the same level as the X. So the mother is the carrier. So mother must have XX chromosomes, 2 X chromosomes. Since she's a carrier, she's actually normal, so she has the big B allele for normal eyesight. And then one small b allele shows that she's a carrier. Okay, so she is heterozygous, having one dominant and one recessive allele for color blindness. Okay, then write down the titles. Parent, father and mother. Phenotype means the observable characteristic. Colorblind for father, carrier for mother. And then genotype is the genetic content or the pair of alleles, all right, like this. For sex-linked trait, it's like this. And then meiosis occurs, so write the process meiosis here. And then gametes are produced. So gametes, for gametes, we draw circles for the gametes, okay? So just separate X small b from Y, correct? And then this X big B from X small b, right? Then during fertilization, any member of the first pair of alleles, or first pair of gametes here, can combine with any of these gametes. So the first possibility is first with first, get X big B, X small b. First with second, first gamete with second gamete, X small b, X small b, okay? Then second with first, these are all the boys, X big B with Y. Always write the X before the Y, okay? And the last one is X small b, Y. Okay, so from the genotype of the child, we can determine the phenotype. Now, the first one is a girl, XX, so she's a girl, and she has the normal allele for normal eyesight, the big B. So she's a normal girl, but she's a carrier because she has the 1 X small b, okay, for color blindness. She carried the allele, but a person may not know that she's carrying the allele, okay, unless they do the genetic test, right? Because they look normal. So the next one is first gamete with second gamete, right? So you get X small b is a colorblind girl because of the 2 recessive alleles for color blindness. Okay. Now then, this is the normal boy, is the normal allele for normal eyesight, and this is a colorblind boy. As long as there's one allele for color blindness in the male or in the boy, is considered colorblind already. Okay, because they only can possess or have one allele for color blindness. The Y chromosome does not carry any allele for this trait, okay, for eyesight or normal eyesight or color blindness. So what's the probability of their child having color blindness? So look at all the children. Which one are colorblind? This girl here and this boy. So it's 2 out of 4 children here are colorblind, and we write it as a percentage. So 2 over 4 times 100, this is a sort of calculation, equals to 50%. All right. Second question. What's the probability of the child having normal eyesight but it's a carrier? So the genotype is X big B, X small b. Okay, so there's only 1 out of 4. So 1 out of 4 children is a carrier. So 1/4 times 100 equals to 25%. Now, if the child born is a boy, what is the probability that he's colorblind? Okay, so if it's a boy already, that means you just consider the boys. Out of 2 boys, 1 is color blind. So the answer will be half times 100 is 50%. If once is determined that it's a boy. Okay, now that's all for this lesson. Hope you have learned something from it, and all the best for your biology exam. Bye for now.